Why don't you start by saying where you're calling in from?
Sure, yeah. And thank you for having me, by the way. So yeah, my name is Josh Atkinson, and I'm calling in from Aarhus, Denmark, where I'm a NSF postdoc fellow working at the Center for Electromicrobiology.
There is a lot to unpack in that one. So I don't know that everybody would know, one, what a postdoc is, or two, what an NSF fellow is. And certainly electrobiology has a lot of interesting qualities to it. So pick and choose. What would you like to unpack first?
Well, I'll go with postdoc because it's a straightforward one to start with. So a postdoc is a type of position that people will oftentimes do after they finish their PhD and do graduate school. And so it allows you to kind of go work in a lab for a period of time, hone your skills or learn new skills that you didn't have during your PhD before you go and maybe go work in industry or try to apply for faculty positions or do whatever you choose to do, because people go down many different paths.
Right. No, that is true. So yeah, I guess I had not, when I was young, appreciated that a postdoc was part of my training too. So yeah. So cool. So what are you doing there, like work-wise?
Yeah, so I'm a synthetic biologist by training, but I'm here in a microbiology department studying organisms called cable bacteria. And so these are these large multicellular bacteria that live in sediments in the bottom of lakes and in the bottom of oceans. And what they do is they can oxidize sulfur down in the anoxic part of the sediment, and then they can transfer electrons centimeter distances away to where there's oxygen up near the pore water in these aqueous systems. Sorry, big words there. But what's really unique about these organisms is that they're very highly conductive. So they have these nanowires in their outer membrane that allow them to transfer these electrons really long distances. So typically when we think of bacteria, we think of short, 1 micron cells that, 10 to the minus 6 meters in size. But these bacteria, you can actually see with your naked eye. You can take a sediment, pull it apart, and see these strings of these cable bacteria growing in there.
What? Like,
Yeah.
I've never heard of this. This is so amazing. So they're not microscopic, and they are like functioning like wires.
Yeah. So there are these kind of high voltage wires that can transfer currents that are similar to the copper wires in your household wiring, but living in sediments all over the globe.
Incredible. Wow.
And so the group,
yeah.
I was just going to say the group here in Denmark are the people who discovered them. So I came out here to work with, founders of this field.
That's amazing. And how long will you be there?
So I'll be here for a year in total. I'm already about 2 months into it. So I'm going to be here until the end of next August.
That's so cool. So I love that you introduced yourself as a synthetic biologist by training, because synthetic biology has not been around all that long. So you might be among the first generation that can introduce yourself as a synthetic biologist by training. So where did you get your synthetic biology training?
Yeah, so I did my PhD at Rice University in, as far as I know, the very first synthetic biology PhD program in the entire country. So I was in the systems, synthetic, and physical biology program at Rice. And I was part of the very first cohort of students to go through that. So yeah, I do think probably one of the first people to be able to say I'm a synthetic biologist by training, yeah.
That's so cool. That's great. And so when you were in college, if you were so early on synthetic biology, what did you study in college that led you to a PhD in synthetic biology?
Yeah, no, I kind of stumbled into synthetic biology a little later. So I did my undergraduate degree in microbiology at the University of Michigan. And I started off getting interested in research, wanting to, go into oncology and cancer research and try to, cure cancer. But I went to go do a research program that was operated by the university, and I paired with a microbiologist where I was studying these bioluminescent bacteria that live in deep sea fishes, which I never had even thought of before going to college. And I learned about all these crazy things that microbes can do and just kind of became obsessed with microbiology there from. And then as I was going through, undergraduate, I was trying to, how can we apply microbiology to make the world a better place? And synthetic biology was kind of rising rapidly at the time that I was doing my undergrad degree. And I heard about iGEM, and I got really interested in that. So I joined the Michigan iGEM team and went to Boston and got to do the big jamboree. And that was really highlight of my undergraduate research career and really drove me into the synthetic biology field. So I started hunting for graduate programs that did synthetic biology, and there weren't very many, but then I saw, actually, I think I got a Google News alert about Rice starting this program. I was like, okay, I got to apply to that one. Yeah.
That's awesome. Oh, how fun. And so lots of highlights from synthetic biology, and you have certainly like touched on some of the most charismatic of systems. I love that, you just described very offhanded and very correctly that, synthetic biology is just using biology to make the world a better place. And you can do that with bioluminescent bacteria, or you can do it with microbes that live in sediments and conduct electricity. There's a lot of biodiversity out there, and to apply it to real world challenges, that's pretty much what synthetic biologists do. So how cool is that? That's great. So let's see. So I'm a little torn, because part of me wants to ask you about your aspirations and where you're going to go next. And then part of me wants to ask you about, when did you get interested in science? So let's ask where you're going to go next, and then, or thinking about going next, and then we'll back it up and ask how you got interested in science at all.
Yeah, so I'm hoping to be able to start my own research group in the future. So I'm applying to faculty positions right now, actually. So it's a busy time at this time of year specifically. I've been prepping, research proposal documents and those kind of things, and sending them out to a bunch of different schools. Yeah, so targeting some in the US, and then also thinking about maybe Denmark as well. I don't know. Yeah, it's nice here.
Yeah. Yeah, despite it getting dark at 4 o'clock, it's a very nice place. Yeah, for sure. So, so were you always interested in science and engineering?
I think so. So it's hard to say. So I got interested in high school for sure. I had really, really good high school teachers in biology, in chemistry, and math. And I think just working with those brilliant teachers and their ability to engage me with these subjects really is what got me interested into them. So I'm the first student in my family to go to college. So I didn't grow up necessarily around a lot of scientists per se. But getting to work with these people at my high school was really pivotal for me. And really, I know my biology teacher in high school. He's the first person who ever really mentioned research to me, because he did some research in his training, and that was like, wow, what is this? And I was like, I need to go do something like this when I get to college.
Wow. You are totally speaking our language. We love our high school biology teachers who are within the BioBuilder community, because they really are inspiring this next generation, and they do just impact people's lives. Right.
I met some good ones. So I was a BioBuilder mentor for the Westboro High team. So Joanne Purdy, shout out to her, because she's a really brilliant teacher.
She is a brilliant teacher. She's actually on our podcast this month. So if you want to catch up with her and hear from her, she's on our podcast this month. So yeah, we love our teachers, and they do impact. They just open possibilities for students that hadn't been necessarily considered before. Where did you go to high school? It's interesting that you're the first in your family to go to college. Where did you go to high school?
Yeah, so I grew up around Flint, Michigan. I went to Flushing High School, which is a town just kind of north of Flint. And so a lot of my family worked in General Motors, or in the automotive industry. A lot of them straight out of high school went and got jobs, either on assembly lines or transporting parts between places. My mom worked in a hospital. That was another very large part of influencing my interest in science. So I got to go visit her at the hospital, and I would go and see some pathologists and get to see some things on slides, and kind of a little sciencey environment. And that's really kind of got me there.
I love that. These little nudges towards science, right? Just shadowing your mom at work at a hospital and seeing a pathology slide is like, oh, science is all around us. And with synthetic biology, those skills of taking things from a manufacturing plant and getting them out into the world and commercializing things. We are at that position now with synthetic biology. We need all kinds of folks who are interested in working in the field. So
absolutely.
Yeah, that's so cool. And so between your mentoring of the Westboro High School team and other work that I know from the EBRC with you, you clearly have an interest in education. Can I just say how much I love the slide deck you put together for EBRC, and maybe have you introduce that as a resource for students?
Oh yeah, thank you. So this was an idea that I worked on with Michael Sheets, who's another member of the EBRC's Student and Postdoc Association. So we've developed this kind of intro to synthetic biology slide deck so that teachers who don't necessarily have training in synthetic biology can start to teach this at all different levels. So we tried to make this really accessible and break it down into a bunch of different kind of conceptual modules that different people can use. So the idea here is to start off giving people an idea of what is synthetic biology, what are the things that have been done with it already to date, what is our design, build, test framework that a lot of synthetic biologists use to try to do their business, and then what are the biomolecular tools that we use to engineer DNA and RNA and proteins, and how can we use these to build metabolic circuits and genetic networks, and then finally, what are the different kind of organisms that we can port these into? So we could, go into bacteria, like I love to work with, but also mammalian tissues, whole animals, plants, and then start actually engineering entire ecosystems at scale. So can we change the entire chemistry of a soil or a sediment to try to, make more productive fields or things like that, or remediate things that are in the environment. And then finally, a bunch of different industrial examples that exist in SynBio. So hopefully people find this to be a really useful tool that they can take into their classrooms. It's all freely available and can be broken apart as needed. Yeah.
Yeah, I think it is an incredible resource. We will be teaching with it as part of the BioBuilder Club, and we will absolutely. It's great because it includes so many examples and covers the range of opportunities within synthetic biology. So it's amazing. Like I feel like I've gotten a whirlwind tour of synthetic biology and the impact that it can have on a person's career. I guess the last thing I'll ask is maybe if you had anything you wish you had known back then that you know now that you would tell students in high school, or sometimes they ask about what they should be doing or thinking or learning.
Yeah, I think being really open-minded is probably the best thing. So I went in, with an idea of what I wanted to do, but along the way, I bumped into all these different topics that have like really broken open my interests. So like I wouldn't have been a microbiologist hadn't I not randomly paired with that person. I'm currently what I would call a microbial electrochemist. So I do a lot of electrochemistry, which was a thing that I had never even thought about before a project that I started doing late in my PhD. So now I'm constantly working with electronic devices and how we can interface cells and proteins with these devices. And, just being willing to go with the flow as things come across your desk and like new ideas. It's really fun to be able to explore those. And I've really enjoyed that kind of aspect of academia because it kind of gives you the freedom to do these things to some degree.
Yeah. Wow. I mean, well, you also seem very brave, right? So some people like get scared to try all those, that freedom is a little bit overwhelming to people sometimes. And people start to sort of go through paths where the ground's already softened and they're aware of what to do and what's expected. So one question that was asked in the Q&A is, as a first-gen college student, do you have any advice for other first-gen college students, especially if they were thinking about advanced degrees?
Yeah, talk to people. So people are very friendly. So I didn't necessarily go to a lot of office hours when I was an undergraduate, and I kind of maybe regret that. I think I could have learned some subjects a little deeper than I did had I gone and had conversations with professors. I kind of thought, oh, I'm going to be a burden if I go talk to this person. I don't need to do that. And I've continually had this come up throughout my career. So as a PhD student, I was constantly, in my advisor's office, just chatting about ideas most of the time, even. And also when you're struggling, they can be really helpful resources. So just being willing to just go talk to people because you're not a burden and people like to help. Yeah.
I think that is fabulous advice. Thank you, Josh. What a great note to open up the conversation to others now. So thank you.